In this video, we'll be again studying about the post-fertilization events, that is the events that are taking place after the process of fertilization. So in the previous video, we have studied about the development of an dicot embryo where the two cotyledons were formed. Now in this, we are going to study about the development of a monocot embryo, and grass is taken as a very common example to study about the development of the monocot embryo. Let us study now. Now here we know the zygote formation has taken place due to the fertilization. And same process, like we need to label the chaliceal end as well as the micropylar end, so to make it easy to understand, and also we'll have a clear picture which part develops into what. Now here the zygote in this case again is a diploid structure due to the fusion of the two gametes, one male and female gamete, and this zygote undergoes transverse division. Transverse division means like this, it will divide, resulting into the formation of three cells. Now in dicot, if you recall, there is formation of two cells, the terminal cell towards the chaliceal end and the micropylar end will have the basal cell. But the development of embryo in case of monocot is different from that of dicot, because here we are having the three cells. So the cell which is towards the micropylar end is named as basal cell, whereas the one which is towards the chaliceal end is named as terminal cell. Now the cell which is a new cell which is present in between the terminal cell and the basal cell is known as the middle cell. So this middle cell is very important because it will later on form the major part of the embryo. That means almost all the structures of the embryo. Now, this basal cell will be studying one by one how the cells are developing different parts of the embryo. The basal cell, which is marked as red over here, in this case, on the monocot embryo, that means in the development of the monocot embryo, the basal cell does not divide, but it enlarges. So this is just a graphical representation to give you an idea what happens. So the basal cell will not divide, but it will enlarge. It will form an enlarged structure. Like we have studied previously, that basal cell will be forming the suspensor and the haustorial cell. Over here, it is not forming so, but it will enlarge and form a structure. The middle cell will divide in all the planes, and as a result, it will form a mass-like structure. So here we see the middle cell dividing and forming a mass-like structure. And when it is not very clear, like when we see from one side, it appears to be a mass. And this middle cell, since it is dividing in all the planes and it forms a group of or a mass of cell, it is the structure which will be forming the plumule. That is what is we call it by other term in the case of monocots. Then it will form the radicle, it will form the epicotyl, hypocotyl, and also the suspensor. So the middle cell is forming the major part of the developing embryo. The terminal cell also divides and it will form only the cotyledon. We understand that dicot have two cotyledons, whereas in the case of monocot embryo, it will be only formation of one cotyledon. Let me put it with blue. So the different colors will make you or will give you a better understanding and the picture of the various parts which are developed from the different cells. So the blue is representing the terminal cell, the black is the middle cell, and the red is the basal cell. Now, let us further study. Now, what happens is this portion which is over here. Now, let me familiarize you with the terms which we are going to use over here. In the case of monocot embryo, and we have taken grass as an example, here the grass will have only one cotyledon. Also, the cotyledon is known as scutellum in the case of grass. It is not called as cotyledon but scutellum. We have used another term over here. And the root cap and radicle are enclosed by a protective sheath which is known as coelureza. So the coelureza is a protective sheath that will be enclosing the root cap and the radicle. That is the part. Another important difference is the shoot tip, which is very young, which is still developing, is protected by again a sheath which is known as coleotile. So coleotile is again a sheath which is present on the young shoot tip, which will prevent the or which will protect the developing young shoot. So here we have a few terms that is known as scutellum, which is the cotyledon, the coelureza, which is covering the radicle and the root cap, and the shoot tip is protected by coleotile. So now let us study how the development from this stage to this stage. This is a young embryo we are talking of. Now here, since there is a lack of space, the cotyledon, that is the terminal cell which forms the scutellum, is pushed out and it will form the scutellum over here, which you can see in the blue. This is the scutellum or the cotyledon. Now the black structure which is over here, this is the embryonal axis. So this structure which we see is the embryonal axis, and over here the basal cell. It remains as it is. There is no change in the basal cell. Now over here we see certain development. A mass of cell is present on the basal cell and it is known as suspensor. The suspensor develops from the terminal cell. Now here the embryonal axis is there and the tip of the portion over here is known as coleoptile. This will develop as shoot later on. So coleoptile is the area which is the protective sheath, because we are talking of a young embryo. So this coleoptile will develop as, it will be covering the developing shoot tip. Here is the developing shoot tip. The portion over here that is below the developing shoot is the epicotyle, and the region over here is known as the lower portion after the where the cotyledon node is present. That portion is known as hypocotyle. Just as we have where the two cotyledons attach, it is known as cotyledonary node, and the above portion is epicotyle and the lower portion is hypocotyle. So here it is the hypocotyle, and over here you can see the covering. This is the tip which is coming out and which will later on develop the radicle or this is the shoot tip. So here there is a protective sheath on top and this is coelureza. So this is, we are talking of the young embryo of a monocot. So the coelureza is present protecting the developing root tip and the root cap. The hypocotyle is present towards the cotyledonary node, that is towards the basal cell. Then hypocotyle is the region which is present above the cotyledonary node and below the developing shoot, and coleotile is the area that covers the shoot. Now later on, when we are talking of a mature embryo, this is a mature embryo. Now as the development takes place, let us make this picture more clear. The basal cell and the suspensor degenerate. So the basal cell will disappear and the suspensor will also disappear. This structure will disappear, resulting into a mature embryo. Now you can see over here very clearly This portion which is developing as a cotyledon. So scutellum is actually the cotyledon which is being pushed, and as a result it separates. So it is the cotyledon which will develop, and here is the sheath that is covering the shoot apex. So this covering over here is the coleoptile. The shoot apex can be prominently seen, and there is a flap-like structure which is known as epiblast. Epiblast is actually the rudimentary second cotyledon, or it is the group of cell that actually develops a flap-like structure and remains rudimentary, and we call it as a second cotyledon, which never develops since it's a monocot embryo. Then we have the root cap over here and the radicle. They both are protected by a sheath which is known as coelurhiza. So this diagram represents a complete mature embryo of a cotyledonous plant.